So good morning, Martin. Thank you for those welcoming remarks, and it's really a pleasure to be here at the University of Michigan Caswell Diabetes Institute. You wonderful hosts, and we really appreciate the collaboration and partnership. And thank you for all of you who are here with us today, for coming. Some of you very long ways, and for all of you who are joining us by Zoom. Thank you for participating. So, for those of you who don't know me, I'm Dr. Robert Levine. Professionally, I was trained as a cardiologist, but in the late 80s, I stepped away from my full-time faculty position in cardiology at the Mount Sinai Medical Center in New York to join my wife, Mary Tyler Moore, and her leadership of the JDRF and efforts that we made together to help find cures for type 1 diabetes and its complications. My task here this morning, my purpose this morning, is to try and put the clinical endpoints workshop in the context of the broader Mary Tyler Moore, vision initiative for you, and start today. Bless you. So this workshop and the consortium that we hope that will follow is part of the broader Mary Tyler Moore Vision Initiative. It is a phase 1 accelerator of that initiative, which you see our roadmap here. And I'll go into a little more details about each one of the elements of the roadmap in a moment. Diabetes had a devastating impact on my wife and in her life, and in particular due to her visual loss from diabetic retinal disease and the severe narrowing of her visual fields from her pan-retinal photocoagulation therapy. So we launched the MTM Vision initiative to honor her and her efforts to help find cures for diabetes and its complications, but also importantly, to focus attention on vision in its multiple meanings to people like her. That is, importantly, the vision that has been lost by millions of people with diabetes-related eye disease, the vision that will be lost despite the advanced treatments available for diabetic retinal disease, and then the vision required of all of us to imagine a cure for vision loss and blindness, and to take those specific actions to find the ways to importantly restore vision and protect the retina from the deleterious effects, of diabetes. And today's work and the work that will follow is an important part of achieving these person-centered goals. Our mission is to be a leader in accelerating the development of methods to preserve, restore, and protect visual function in people with diabetes, in particular and including, importantly, those who already have significant visual loss for which there is an important need, as evidenced by my experience with Mary and her loss of vision and millions of others. Our approach is to act as a catalyst by providing what we call accessible resources, or convenings like this, providing the community with critical resources linked to collaborative research networks who are willing to share their data in real time. So back to our roadmap. So phase 1, we call an accelerator. It responds to what we believe are 3 fundamental barriers to accelerated progress. First, you can't solve a problem you haven't defined. Second, you can't find cures for human disease without studying the human condition. And third, you can't judge success unless you know what to measure. So the response to the first challenge, you can't solve a problem you haven't defined, is our ongoing DRD staging update project, which many of you are participating in and which informs our work here today. In that project, we've tasked ourselves with developing an updated multidimensional DRD staging system, which can incorporate measures of visual function and retinal physiology, the molecular milieu, whether it's by blood samples or aqueous or vitreous humor evaluation, systemic factors, and importantly, bringing the patient voice into the picture in order to better diagnose the disease earlier in its progression, better define its severity, predict and measure response to therapy, and support clinical trials. And a natural output of that process, that project, has been the identification of promising biomarkers and clinical endpoints that need further development and validation, which again is the subject matter for today's work. Our response to the second challenge, you can't find cures for human disease without studying the human condition, is standing up of an ocular biorepository here at the University of Michigan under the direction of Patrice Fort. Now, we've modeled that against the highly successful and game-changing JDRF network for pancreatic organ donors with diabetes, which has changed the way in which we understand type 1 and type 2 diabetes in humans. We will be collecting and characterizing cadaveric samples under a special protocol developed by Dr. Ford in order to ensure that we are able to do the detailed omic analysis of the samples to better understand the cellular and molecular markers associated with progression of diabetic retinal disease and identify targets for therapeutics development. In itself, that's not enough. So this will be all put on a data sharing platform, a sample and data sharing platform, which will be available to the academic community as well as hopefully form the basis for an industry consortium to accelerate our progress. Interestingly, Dr. Ford has also acquired a unique imaging tool, which allows him to in sync do a color photograph of the retina as well as an OCT. So we can now map the OCT to the color photograph to get a better understanding of the disease. And then today, we're in the kind of the answer to the third challenge, which is you can't judge success unless you know what to measure. That's what the whole point of this clinical endpoints workshop is and the consortium that will follow, which is to start to look at what we call, at least one of our goals, to identify a basket of promising clinical endpoints, including biomarkers, imaging methods, functional assessments, and PROs that are first useful, and then second, may have potential to become registrable endpoints individually or as a composite, and plan the trials that are necessary to start to learn more about the potential for those endpoints. Just looking at the other two phases of our roadmap quickly, we move from these fundamental accelerators that we call them to development of advanced diagnostics and therapeutics target identification. The first stand-up project of that will be a retinal imaging core, which will try to bring together millions of images associated with longitudinal clinical records and functional assessment to try and get some signal out of it using AI tools for better diagnosis and prediction of course, and then of course modeling the successful Michigan kidney translation core here. Our intention is to develop yet another consortium that can look towards what we would call a therapeutics target identification core to kind of serve the global academic community in precision medicine, but also establish support for a pre-competitive consortium of industry to, if we call it de-risking the space to incentivize investment in DRD indications. And then last is our science with a mission, programs which will focus on, milestone-driven science to preserve, importantly, to restore and protect vision. And those may be retina replacement through ex vivo genesis of a neoretina for implantation, or learning how to induce regeneration with stem cell therapies, tissue transfection, or gene therapies, or looking directly at retinal neuronal cell protection or preservation of vascular integrity, and including reperfusion of the diseased retina. So as I said, in the context of the broader Mary Tyler Moore Vision Initiative, today's effort and the consortium that will follow it to identify the clinical endpoints that we can use in a basket of endpoints to better define the disease, including the patient voice, is a critical part of our achieving our broader goals, as you likely recognize from the discussion and your experience. And finally, just to say a word about why we do the things we do. All of us have a choice in life. We can either kind of float with the tide or swim to a goal. We choose to swim. Thank you very much.